Good morning, all. So today I'll talk about different AFA-assisted methods that we are exploring and using for high throughput mass spectrometry-based proteomic analysis of FFP samples. So we are all familiar with the advantages of using FFP samples for proteomic analysis. These are abundant archival samples with cost effectiveness in terms of storage. And these are usually present with rich clinical annotation, so adding clinical relevance to the study, enabling retrospective analysis and careful selection of samples. And it is also a sample of choice for proteomic analysis in spatial manner. In our lab, we have a number of projects where we are using FFP samples for proteomic analysis, and we are using a variety of methods for processing these samples. A while ago, this is the method that we were using for analyzing FFP samples. We collect the tissue in a microfiche tube by laser capture microdissection, followed by decross-linking for 1 hour at high temperature. This was followed by reduction alkylation and overnight digestion with trypsin. Following day, samples were acidified and transferred to an autosampler vial and analyzed on mass spectrometer. So some of the limitations with this method is that it is a time-consuming method with overnight digestion, and it has limited scalability. It includes a lot of manual steps with limited potential to automation, and thus it's not a method of choice for high throughput projects. So we wanted to explore new methods and improve this method to make it a high throughput method and also reduce on the turnover. So as Deb in his introduction talk has already mentioned about the benefits of AFA, how it is useful in terms of reproducibility, sensitivity. So in our lab, we are using a Covari's 230 ultrasonicator instrument. The two added advantages we have with this instrument is that it is compatible. It is compatible with both strip tubes as well as a 96 well plate. So adaptable to less number of samples as well as a high throughput project. Another advantage is that it has a water bath which maintains the temperature, thus maintaining the uniformity across the samples, resulting in reproducibility. So our sample of choice is FFP tissue captured by laser capture microdissection, and the instrument, the Leica instrument that we have in our lab for LCM, it has a holder that specifically fits, that is designed for the AFA compatible 96 well plate. So instead of collecting the sample in a centrifuge tube, we can directly collect a sample in the 96 well plate, making this method suitable for large number of sample and avoiding the transfer of the samples, as the processing can happen directly in the 96 well plate. So with all these advantages in mind, we were curious and interested in trying addition of AFA to our current workflow. So this is the experiment that we designed, and Dave has already mentioned about this in his talk. We had a publication from these results. So for this, we collected 20 square area in 3 sets of 16 samples each from a single FFP block. The samples, as I mentioned, were collected in 3 sets. One set was collected in microfuge tubes, and in another 2 sets, they were collected in AFA-compatible 96 well plates. The samples that were collected in tubes were processed as following the standard protocol that we had developed in our lab, while the samples that were collected in 96 well tube were subjected to AFA treatment before and after the decross-linking step. The difference between the two steps following the decross-linking was that one of the plate was subjected to overnight digestion at 37 degrees Celsius, while the another plate was subjected to a 1-hour digestion with application of AFA during digestion. Following digestion, samples were subjected to desalting, followed by analysis on an Orbitrap instrument. So these are the results. We could identify more than 1,000 proteins across the 16 samples using the standard method. These numbers were slightly improved in the AFA accelerated method, where the numbers were close to 1,500. And as we can see, the identifications were reproducible across the 16 replicates that we have used. This table mentions the average number of proteins and peptides that were identified across the 3 methods. The methods were also comparable in terms of the miscleavage that were identified across the samples using the 3 methods. There was a good overlap that was observed between the identifications of protein and peptide across the 3 methods, showing that AFA accelerated method is comparable to the standard method as well as it is reproducible. So in the previous experiment, we have used a larger area, that is 20 mm square. We have also tried the same method going down on the input sample. So we have also tried an area of 1 mm square as well as 0.0 mm square, where the expected yield is less than 1 microgram of total peptide. And these are the numbers of protein and peptides identified across the 2 sets of samples. With the 0.5 mm square area, we were able to detect close to 1,000 proteins. And with 1 mm square, we were able to identify close to 2,000 proteins. As we can see here, the numbers were reproducible across the different replicates that were used. So, so far the experiments we have done were based, there the sample collection was based on the areas. The recent round of experiments that we are doing, here we are collecting samples by the number of cells. So we have collected 10 cells, 50 cells, and 100 cells. And instead of collecting these cells in a 96 well plate, we are now collecting them in a 384 well plate, which is AFA compatible. So to test this method on the lower input samples, we have extracted samples from the colon tissue in a 384 well plate, and during the lysis and digestion, we have used AFA application, and this is a 60-minute digestion. Following digestion, the data was analyzed on a timsTOF ultra-true mass spectrometer in DDA passive mode. And these are the number of proteins and peptides identified across the 10, 50, and 100 cells. In 100 cells, we were able to identify close to 4,000 proteins, and then these numbers were close to 2,000 in 10 cells. And these are the number for the peptides identified in 100 cells, close to 20,000. In 10 cells, close to 10,000 peptides were identified. The error bar here represents the standard deviation across 3 replicates. So we can see that we get reproducible results with AFA-assisted method. We also analyzed the peptide length of the peptides that were identified across these samples. Across the 3 set of samples, the peptide length, most of the peptides were 7 to 12 amino acid long in length. We have also used this method for analysis of single cell. The data looks good along the single cell as well, with close to thousands proteins being identified and close to 4,000 peptides being identified. However, we are still working on this experiment in terms of reproducibility. So the advantage here is that we have a shorter method with only 1 hour of digestion time. We are able to collect the cells directly in a 96 well plate or a 384 well plate, and we can directly process the sample in the plate that we have collected, and also the sample can be injected from the same plate. So there, we are avoiding the losses that could happen due to transfer of samples from one vessel to another. So here we are able to gain the sensitivity as well as reproducibility, and also because we are performing the process in a 384 well plate, it's a high throughput method. Another experiment that we are doing with AFA application. So we have a project where we have close to 150 samples, but here we have to analyze, instead of tissue sections, we have to analyze tissue punches, where the tissue is collected from an FFP block in form of punches. So here we are using bead snap cap, which is AFA compatible small tubes with a homogenizing bead with a glass bead in the tubes. So we can use these tubes for homogenizing the tissue, and these tubes we can use either in a 96 well plate format or as a single tube. So again, we have a choice of number of samples we want to process at a time. So the experiment that we designed for this analysis is that we collected 1 by 1 mm tissue punches from a FFP block that was made from the cerebral cortex tissue. And after collecting the tissue, following deparaffinization, for homogenization, we have tested either the probe sonicator or the AFA method using the bead snap cap tubes. So the numbers of proteins and peptides that we identified by either method were comparable. However, the added advantage with the AFA method is that probe sonicator is a manual method, and also the temperature during the sonication is not controlled, which could introduce variability from sample to sample analysis. Using the R230 ultrasonicator, we have a control on the temperature, so we don't need to worry about the reproducibility across the sample, and also we can process 96 samples at a time, reducing the manual time. This is the overlap of the identification, showing that homogenization with the AFA assisted method works equally well as the probe sonicator method. And in terms of the peptide length identification also, both the methods work similar. And in terms of miscleavage also, the two methods were comparable. So summarizing our work, we have seen that we are able to get reproducible and sensitive data using AFA-assisted method for FFP analysis. Also, these methods have potential for high throughput as well as automation. Finally, I would like to acknowledge people from our lab, Dr. Akhilesh Pandey, Chris, who has worked on sample preparation for these projects, and Amy J. French. Chris and Amy, they both helped in sample collection by LCM, and Benjamin Madden, who helped in generation of some of the earlier data that I presented from the 20 mm square area samples, and also the Covaris team. This is our group at Mayo Clinic.